Have a very good morning, everyone. We're very pleased to have with us our Secretary General for the university, Eduardo Bárcena Garcia, in our chair today. We also have Dr. Federico Sanchez with us, our head organizer for the event. And also good morning to all other distinguished members of our community, particularly the heads of our chemistry faculty and the Institute for Biotechnology. Also, before we begin, we would like to thank everyone who has supported us to make this event happen. From every student who had something to tell us on how to make things better, to every professor who had advice for us, whether it was financial, from logistics, or academic. Without you, this wouldn't have been possible. Thank you to our friends, and thank you to everyone who funded us. You really have made a change in our lives. As I stand before you, I feel a thrill and joy similar to that of when, at the age of 3, I gained my first notion regarding the history of life on Earth. I did it by watching Walt Disney's Fantasia. The animated interpretation of Igor Stravinsky's The Rite of Spring captured every bit of the drama, the violence, and dynamism needed to fuel a passion which led me first to watch the movie countless times, and later on, to search for a better understanding of that very same story, one encompassing mystery, science, life, death, competition, because we have all been stricken by that same lightning, upon which many great minds have built their careers. We're brought together by our respect and curiosity for a force chronometered in geological seconds, minutes, or hours. It's also both molecularly small and population-wide, a force so irresistible that even our study of it has suffered transitions and changes.
It was 20 years ago, two evolutionary biologists decided to publish a book which offered a vision of evolution unlike any other seen before, Dr. Eros Somari and Dr. John Maynard Smith. Since then, the biological sciences have suffered also several major transitions, including the dawn of the genomic era and the post-genomic era. Thank you very much for being here, and welcome.
Well, let me tell you the truth. The researchers are them. They and many other students, undergraduates mostly, who put their passion for evolution, for science and genomics. And the original idea, and just give me the first one, the first slide, please. I would like to start just to tell you that this is, well, not only welcome to U.N.A.M, but welcome to Mexico, because this event is being transmitted on site, real time, everywhere. And this is one of the reasons that Dr. Bárcena was very kind to offer this place, because we have a very good streaming. So everybody in the metro or in the underground, wherever people is located in Mexico, can see this event in real time. So this is one of the major reasons that, of course, we are U.N.A.M, and we are very proud to be here. And the other truth is that this idea started about 2 years ago. And how did it happen? Well, it happened that I was in Seville, in December 2013, and I had the chance to hear Iñaki talk in the faculty in the School of about the origin of multicellularity in animals. And this was a theme that had not been covered in our undergraduate program in genomic sciences. So I became very excited, and when I came back from Sevilla, and I talked to Juan and Bere, and I told them, because the Bere was my course assistant, and Juan was one of my students in this course. And so I told them that I was very excited with this new theme, and I said, no, but this is part of the major transitions in evolution, and we should cover all of them. Because why? As Bere has already told you, 20 years have passed already from the publication, the first publication of this book, this seminal book, in which I use for the first 4 or 5 classes in my course in cell biology. Of course, your book too is, Bruce, is part of the central core of the course. And then the students took from their own heart the organization. They wrote the letters to you, because probably you were surprised that the letter was written by an undergraduate student asking you to come to Mexico to give a lecture and to participate in the symposium on the major transitions and evolution. The students were the ones that made it. They also wrote the grants for the council, Research Council and Technology in Mexico. They did most of everything. Even this was printed by them. So really the heroes are our students, mostly from the undergraduate program in genomic sciences, but also other graduate programs, and also in other parts of Mexico. So with no further explanations, I would like to invite you Dr. Eduardo Bárcena, who is the General Secretary of U.N.A.M, to address you for the opening.
Well, good morning, everybody. Thank you very much, Federico, and this great team that organized this major event. I was asked to give you a brief introduction and a brief opening of this ceremony, but I wasn't asked to do that in English, so I prepared it in Spanish, and I will read it like that. So, distinguidos académicos, nos acompañan apreciables invitados, señoras y señores, amigos todos. Es un honor para nuestra casa de estudios el recibir a todos ustedes en este importante recinto, destinado, entre muchos otros aspectos, a albergar eventos de gran trascendencia como el que hoy nos convoca. Es pues un orgullo para nosotros que el Centro de Convenciones y Exposiciones de la U.N.A.M, referente y medio de vinculación con la sociedad mexicana y el mundo académico, se constituya en la sede del Simposio Internacional de Major Transitions in Evolution. La U.N.A.M acoge hoy la distinguida presencia y participación de profesores extraordinarios, académicos del más alto nivel, investigadores laureados con los máximos reconocimientos internacionales en su área y premios Nobel. Nos visitan insignes especialistas provenientes de las más prestigiadas instituciones académicas de Alemania, España, Estados Unidos, Canadá, Francia, Gran Bretaña, Israel, Italia, Portugal y desde luego de México. A todos y todas, nuestra casa de estudios les abre las puertas, les brinda la más calurosa de las bienvenidas y les agradece su intervención en este magno evento con el que se refrenda la visión de la UNAM para congregar en su seno a distinguidos exponentes académicos para la discusión, el intercambio y la reflexión sobre temas de la más alta relevancia a la altura de las exigencias de nuestros tiempos. Gracias nuevamente por darle con su presencia el sentido y la dimensión trascendentes que con ella adquiere este simposio. Nuestro reconocimiento también al Instituto de Biotecnología y al Centro de Ciencias Genómicas de la UNAM, a sus autoridades y a los organizadores por hacer posible la realización de este evento de magnitud internacional. Tomo unos minutos de su tiempo para una breve reflexión relacionada con la temática que hoy nos convoca. En nombre de este simposio, que a su vez es título del libro The Major Transitions in Evolution de John Maynard Smith y Eros Samartmary, hace pensar que el evolucionismo también evoluciona. Ciertamente corresponderá a ustedes, distinguidos especialistas, el análisis sobre los nuevos paradigmas que se gestan en la biología estructural e integrativa, en bioinformática y biogenética, en física, lingüística computacional, teorías del aprendizaje, historia y filosofía de la ciencia, en genómica y bioquímica o biología molecular, en embriogénesis, arqueología y células madre, entre otras. Sin duda, el surgimiento y desarrollo de las teorías evolucionistas han tenido un gran impacto no solo del modo en el que hoy entendemos la docencia y la investigación, sino también en la manera en que asumimos y concebimos a la universidad del siglo XXI. Todos sabemos que desde su aparición y hasta el día de hoy, la teoría evolucionista ha despertado y sigue provocando apasionados debates y controversias que no se limitan al campo específico de la biología, sino que se extienden al ámbito de la filosofía, la ética, de las leyes y de la moral, entre otros. En este sentido, es posible afirmar inequívocamente que el evolucionismo nos atañe a todos, en muchos sentidos y de varias maneras. Recordemos que desde el famoso debate de 1860 en Oxford, pasando por los diversos juicios norteamericanos del siglo XX o la prohibición de su enseñanza en el siglo XXI, lo que Darwin denominó el misterio de los misterios, es decir, el evolucionismo, ha sido continuamente cuestionado, objetado, censurado e incluso condenado a la usanza inquisitorial. Los 156 años de polémicas desatadas a partir de su aparición dan cuenta del enorme poder y trascendencia de esta teoría, considerada desde sus inicios como una idea peligrosa. Las contribuciones de Darwin, invaluables para la biología y muchas otras ramas de las ciencias y las humanidades, trascienden, insisto, el ámbito exclusivo de su disciplina. En su contexto histórico, el trabajo del naturalista inglés dejó en claro la necesidad de separar ciencia y religión, y por ello, uno de los aspectos más importantes de su legado consiste en el establecimiento del laicismo como una condición sine qua non para explicar la realidad. Así es como la teoría evolucionista se convierte en un auténtico parteaguas fundacional, una verdadera revolución intelectual en la búsqueda del conocimiento que se hace patente en nuestra manera de entender la educación universitaria de hoy. La UNAM tiene así entre los pilares que sustentan su valor el principio irrenunciable de la laicidad. Esto en congruencia con su origen etimológico griego, laicos, que significa del pueblo, y del latín laicus, el que no tiene órdenes clericales. Esto es un valor supremo, un componente indisociable de la vida democrática y una condición irrenunciable para el avance del país. La gran virtud de este valor en el ámbito universitario es que garantiza mecanismos de análisis, entendimiento e interpretación libres de toda visión sobrenatural. Como institución pública, educativa y laica, la nuestra asume una postura universalista de respeto al pensamiento individual. Entiende y asume el laicismo en todas sus prácticas cotidianas como garantía de los derechos humanos fundamentales, como condición de la vida democrática y como componente de nuestra soberanía. Lo concibe como el valor indeclinable que asegura que la docencia, la investigación y la difusión de los beneficios de la cultura sean ejercidas en toda libertad y con absoluta responsabilidad social. Por eso, el dogma y la verdad unívoca no tienen lugar en la UNAM. Aquí aprendemos a descifrar e interpretar el universo en todas sus dimensiones, a partir de la incertidumbre, de la duda, de las interrogantes que sobre él tenemos. Queremos saber, no creer. Estamos convencidos que el único camino hacia la construcción de una mejor sociedad y de la realización personal de los individuos que la integran es el conocimiento, concebido como herramienta de transformación en oposición al fundamentalismo sustentado en la ignorancia e invariablemente orientado al control ideológico y al uso del poder. Por eso asumimos que la laicidad, el saber y la democracia constituyen un trinomio indisoluble, una condición para el bienestar humano en su más amplio sentido. Ese es el significado y esa la dimensión que, amén de los saberes específicos y especializados que habrán de debatirse, adquiere el simposio cuyos trabajos dan inicio el día de hoy aquí en la Universidad Nacional Autónoma de México. Reitero pues mi gratitud a todos y cada uno de los que con su participación en este evento, sea organizativa o argumentativa, propician el avance del conocimiento al servicio del bienestar humano y contribuyen con este ejercicio académico a consolidar el rol que la universidad desempeña en las grandes transformaciones de la sociedad. Muchas gracias. Bueno, y con la autorización de ustedes, siendo las 9:05 del lunes 23 de marzo de 2015, en el Centro de Exposiciones y Congresos de la UNAM, en nombre del señor rector, doctor José Narro Robles, y el mío propio, declaro formalmente inaugurados los trabajos del Simposio Internacional de Major Transitions in Evolution. Que salga todo muy bien. Muchas felicidades.
Just as a reminder, after each conference, we will have a discussion, and we have the pleasure to have moderating these discussions Dr. Rosie Redfield. So you will get to know her soon, and we hope that this provides another setup that can enrich our conferences. So now we finally begin. Thank you.
Thanks everyone for your attention. Well, welcome to the first session of the Major Transition in Evolution meeting. It's good to begin at the beginning, in this circumstance, the beginning of life. Let me introduce the first presentation. We have two wonderful talks to start, the origin of life and the RNA war by Professor Lascano, and also origin and evolution of ribosome by Professor U.N.A.M. As you know, early evolution of life is a long period of time between the origin of life to the first eukaryotic cell, from around 4 million years ago to 2.4 billion years ago. Standard period with a lot of unanswered questions. Interesting but unanswered questions, many of them. For example, how did life appear? What was the first genetic material? How did evolve to DNA? Which were the first metabolic pathway? How were assembled? When and how the eukaryotic cell evolved? And many more queries. But today, we will focus in a short period of time. The epoch where life was originated, the epoch with the RNA war evolved from, not working, but from RNA war to RNA protein war to DNA and RNA ribosome protein life. Fortunately, in the last decades, the important developments of the study of these areas have produced significant results in both approach, the synthetic approach and the historical approach to early life. The synthetic approach and the historical reconstruction give us, for example, a robustness of the RNA war hypothesis, the self-sustained replication of RNA molecules, the RNA ribosomal catalyzed peptide bond formation, and ribosomes that catalyze metabolic reaction. But in the other direction, in the reconstruction of life on early stage, we have also a set of results. For example, we know that the set of most highly conserved sequence common to all extant genome includes a gene from different evolutionary stage, for example, from a ribosomal protein, maybe from the RNA protein war stage, but also some transferred from DNA, RNA proteins war, but maybe the for this session, a significant majority of genes evolve in RNA metabolism from the RNA protein war. But today, we are privileged to have two international authorities on the field. Our first speaker, Professor Antonio Lascano, is one of the most recognized Mexican scientists in the world. He was twice president of the International Society for the Study of the Origin of Life. He was the first Latin American scientist to have had this position. He has studied the origin and early evolution of life over 40 years and is the author of several books, including The Origin of Life, with over 800,000 copies sold. And his current research interested in the chemistry and the origin of life and the use of phylogenetic database to reconstruct the early stage of cell evolution. He has just been appointed member of the El Colegio Nacional, the most important Mexican honorary academy. It is with great privilege to present to you my professor, my colleagues, and good friend, Antonio Lascano.
Well, good morning. Thank you very much. I want, first of all, to thank very deeply, very sincerely, the organizers of this meeting. I am very moved, and that's the only word I can really can really describe properly my feelings. The engagement and the work that was done by Berenice Jimenez, ladies go first, by Juan Escalona, Don Juan, and by Federico Sanchez, my very dear friends, but most especially, and I think that already Federico Sanchez addressed that issue, I'm very, very moved by the extraordinary effort that two students of our university did in order to organize this meeting. Having been organizer or co-organizer of a number of international meetings, I know the troubles that one has to go through, and it's really impressive to see how successful they were in convening people from all over the world, in getting the resources, and the easiest part, of course, was to have here students from our university, because as Dr. Bárcena already said, this is a university in which we do not have any problems teaching, promoting, discussing the evolutionary ideas. The other thing that I want to say from the very beginning is to thank my dear friend and colleague Arturo Becerra for his very kind introduction. And secondly, some of you may have wondered, as I did, why my name is twice in the program. And that's for a very simple reason. I have the honor of substituting Sidney Altman, who has a Nobel Award. I don't, because of his discovery of the catalytic properties of RNA, but he was unable to be here. Unfortunately, he canceled under the very last moment, so the organizers were faced with the problem of having to fill up the slot, and that actually gives me the opportunity, if not to substitute him in full, at the very least to actually address the issue of the origins of life and the RNA world. Now, don't worry if you cannot see clearly what's in the screen. The origin of life is indeed very difficult to understand and to see. So some of the things are going to be actually a little bit hazy, not only because of the light conditions, but also especially because there are some aspects of chemistry and of the conditions of the primitive Earth, the nature of the first living entities, which we simply do not know very little about. So if we can start with the first slide, let me begin by saying that it is generally said, mistakenly, that the origin of life is a problem that has been of interest to philosophers, scientists, society in general, since many, many centuries, since many millennia ago. And this is not really the case. For a long period of time, for many millennia, actually what the people thought as a spontaneous generation was simply a non-sexual way of reproducing. And it is not until the 19th century when we have this extraordinary development of evolutionary thought that, in fact, the idea of the origin of life took the meaning that it has nowadays as referring to the start of the biosphere, as referring to the appearance of the first living entities. And one normally relates this development of evolutionary thought, rightly so, to Darwin's contributions, but it is absolutely fascinating to look at the intellectual and scientific atmosphere during the 19th century, especially in the Western world, because what we see is that, in fact, the first to speak about the idea of a system, a natural system evolving, were not the biologists, but were the astronomers. Actually, it was Kant and Laplace in the very late, towards the end of the 18th century, that proposed that the solar system could be described not only in terms of the Newtonian equations, which happen to be symmetrical with respect to time, that is, they will tell anything about the actual origin of the solar system, but they came up with the idea independently, Kant and Laplace, that actually a cloud of interstellar material collapsed, and that gave rise to the sun and the planets revolving around this primitive sun. That simple idea is introducing the element time, the component time, in the description of a natural system. And this was followed very rapidly by Malthus, who has a very bad reputation among my most leftist friends. But in fact, if you read Malthus carefully, you realize very quickly that he was describing the evolution of demography of a society, that he was addressing the issue of the evolution of technology. So once again, we have the idea of time, changes in time taking place in this case, in a social issue. Then, of course, we had in 1809 the publication by Lamarck of the Philosophie zoologique, and which can be seen as the birth certificate of Darwin, because Darwin was born precisely that year. And Lyell in 1830 was actually addressing the issue of how to extrapolate from the current phenomena that we see on the surface of the Earth in order to understand the evolution of the planet itself. Sadiq Carnot and Thomson, who were physicists, were actually describing how in an adiabatic system, one that is not exchanging matter and energy with the environment, you see the increase of entropy. And once you have the increase of entropy in a system, we are again saying there are changes that are occurring in time. Languages were discussed by Spencer, the evolution of languages in 1857. Of course, Marx and Engels were discussing in a very teleological manner the evolution of production and hence the evolution of society. Renan was discussing the evolution of languages, of religion. So when we look at it from a broad perspective, really it appears that Darwin is not really the starting figure of the idea of evolution, but represents in many ways, epitomizes in very way the culmination of evolutionary thought. Now, it's very well known that Darwin did not address in public the issue of the origins of life, but he, especially in The Origin of a Species, published in 1859, he barely touches in a very indirect way the idea of how life may have originated. But if one goes to his notebooks, they are absolutely fascinating, because even though he never explicitly says anything about the origins of life, except for a couple of letters that are very well known, in his notebooks, there is this fascinating page that I love, because it has a drawing that summarizes what should be seen as a lesson whenever we are addressing evolutionary issues. Darwin drew here 3 bold lines that represent 3 hypothetical species, and he says on his notebooks that if you compare the physiological traits, the anatomy, the ecological distribution, the behavior of these species, then you can come out with a phylogeny that will say, in this particular case, that these 2 species are descendants of the one that they separated from the ancestor of this other one. And what I like especially about this drawing are 2 features. One of them, the fact that it shows that all of these 3 species come from a common origin, which is really what he provided us with the framework for assuming that all life on Earth has a single origin. All of us come from a single ancestral population. And secondly, and this is a lesson to be kept in mind whenever we are discussing evolutionary scheme, evolutionary scenarios, you see there are broken lines. And broken lines always refer to a hypothesis. Our hypothesis on the origins of life, on the evolution of cells, on the evolution of languages, on the evolution of humankind are always hypotheses. And of course, we refine those hypotheses in a highly eclectic way, taking material or taking evidence that comes from a widely assortment of different fields. But it's a lesson to be kept in mind. And it's a lesson that's especially important whenever we try to go back into the origins of life itself. Now, one thing that is absolutely fascinating about this evolutionary tree is that it's just a hypothetical, represents a hypothetical scheme of 3 species. And of course, thanks to the development in bioinformatics, molecular biology, by the right choosing of a proper phylogenetic markers since the late 20th century, from 1977 onwards, we can construct trees that are in fact very, very refined. I will not address the issue of how these trees are made. This will be discussed by my good friend Simonetta Gribaldo in a later lecture. But we know that the famous distinction between plants and animals is clearly not the most significant one when we go into the molecular, when we choose a molecular marker to make this kind of phylogenies. Here we have 3 major domains, as some people will call it: the bacteria, the archaea, and the eukaryotic nucleocytoplasm. But the point that was already mentioned by Arturo Becerra in his kind introduction, and that will be addressed by my friend and colleague Luis de Lalle, is that just by looking at the traits present in all extant living beings, we can come up with a very reasonable characterization of the nature of the less common ancestor. Some people call it Luca, some call it the less common ancestor, some people call it the less ancestral population, whatever. But the fact is that all living beings that we are aware of, all extant life forms that we know, that we have studied, actually are descendants of an ancestral population. But the point that I want to make, that I really want to underline, is that if we want to look at the actual origins of life, we actually have to go further down in time. In other words, the origins of life and the nature of the less common ancestor are completely separate issues. The less common ancestor is separated, if not in time, at least in levels of complexity, by the origins of life, which took place several meters downwards in this direction. So one has to be very careful about extrapolating traits that we see in living beings today and try to assume they were present on the origins of life. Now, whenever we try to do this, we face immediately a major issue, namely that if we go back in time, we know that life is a very ancient phenomenon on Earth. There is some discussion about the actual nature of the first fossils that we can assign to actual biological activity. But most of us, not everyone, but most of us will say that if we compare, for instance, isotopic fractionation, we can be almost sure that about 3.5 to the 9 years ago, there was already a highly diverse microbiota inhabiting the Earth. Now, the problem is that the Earth originated 4.6 to the 9 years ago, more or less, take 100 million years, more or less, take or give 100 million years. And at the beginning, the Earth had very harsh conditions. The conditions of the primitive environment were extremely complicated. There was no land as we understand it now. The surface of the planet was too hot, so we could not have a hydrosphere. There was an intense amount of collisions from infalling material like meteorites, asteroids, comets, and so on. So it's very unlikely that at the very beginning, during the first 100 millions of years of the history of the Earth, the environment was amenable to the synthesis of organic compounds and the appearance of life. So here we have a gap, this question mark, that please keep it in mind because it will appear all along my lecture, all along my talk. You will see this question mark either explicitly or implicitly because we simply do not know how life appears. We really are in a void when it comes to geology, and if we don't have any geological sediments, if we don't have any geological evidence of the conditions of the primitive Earth, it's very difficult to say something to be very sure about, for instance, the pH of the primitive oceans, the composition of the atmosphere, the temperature of the Earth, and so on. In other words, some of the issues that we face when we are trying to understand the origins of life have to do with the lack of geological evidence of prebiotic environment. We don't have a fossil record of prebiological systems. The first fossils that we can identify as such already correspond to very sophisticated morphologies, are evidence of highly sophisticated metabolism. So we really have no idea what preceded those primitive entities, those ancestral entities. And finally, and this is a problem I think that will be addressed by Professor Evelyn Fox-Keller, we do not have an all-embracing, generally agreed definition of life. I am one of those, and I'm very happy to say it openly, I am one of those that is convinced that we do not need a genetic code to have life started, as long as we have a biologically encoded catalyst like RNA, ribozymes, or something equivalent. But not everybody agrees with my viewpoint, and I have to say very explicitly that this is a source of major debates that actually transcend the scientific discussions and touch quite directly issues like what is life, abortion, euthanasia, and so on. So clearly, the lack of a generally agreed upon definition of what life is limiting our understanding of which moment in the history of the Earth, in the history, has started the history of the biosphere. In spite of that, it's fascinating when you look at it from a historical perspective that in the late 20s and then in 1936 in Russian, in 1938 in English, Alexander Ivanovich Oparin, who was a biochemist, one could call him a a Soviet biochemist who actually visited the university, our university, and received an honorary degree in 1979. He proposed an evolutionary perspective on how to approach the origins of life. Oparin's idea was that it was impossible to assume, if you were a Darwinian, that life started right away on a single step. So he decided to divide the issue in a number of discrete steps, and furthermore, Oparin did something that was quite extraordinary at the time, and it was already referred to in a way by Dr. Bárcena. He couldn't care less about the borders separating different fields. He had been trained as a plant biochemist, but he immediately began speaking to astronomers, geologists, physicists, chemists, and so on, and decided that an autotrophic photosynthetic metabolism was simply too complicated, and it was impossible to assume that from the very beginning life had started as being photosynthetic. So if you are not photosynthetic, if you are not an autotroph, you have to be a heterotroph, and of course the most common simple form of heterotrophy is fermentation. Fermentation requires the prior presence, the prior accumulation of organic compounds. Organic compounds are destroyed by free oxygen. So then Oparin made the following sequence of conclusions. If you don't have, if you are to have organic compounds, you cannot have oxygen. If you don't have light, you will not have you wouldn't have photosynthesis. If you don't have photosynthesis, you wouldn't have free oxygen. Hence, the primitive atmosphere was highly reducing. And this was a major conclusion from a biological perspective that was in fact fully supported by what the astronomers were already saying in the 1920s, namely that the primitive Earth was formed in a highly reducing environment, and therefore most likely the atmosphere was lacking free oxygen. If you accept this idea, Oparin immediately went to see not only how you can stabilize, how you can accumulate organic molecules, but also how can you synthesize them. And there was an extraordinary tradition, scientific tradition, coming mostly from the German chemists of the 19th century that had demonstrated the synthesis of urea, amino acids, sugars, and so on. So Oparin came to the conclusion that these organic compounds accumulated in the primitive Earth, in the primitive oceans. If you don't see them clearly, believe me, there were some organic synthesis in the primitive Earth. And then, since he was strongly reluctant to reduce life to one single molecule, he came to the conclusion that quasar-based, which can be seen as the avant-garde model, like le dernier cri de la mode, if you wish, of what the protoplasm was at the time, he came to the conclusion that in fact life had evolved from quasar-based, and from this quasar-based finally you have the first heterotrophic, anaerobic, fermentative bacteria, the first forms of life. Now, if you look at this scheme, one thing that is quite striking is the absence of nucleic acids. But it's quite striking to us because in 1936, when Oparin published his book in Russian, James Watson was in the 5th grade of grammar school. So there was no way that people could think of DNA or RNA being a component of the basic definition of biological activities. Now, we have changed this scheme in a way as shown in the next slide, in which we accept in fact the fact that there was a synthesis of organic compounds. These compounds did not necessarily actually form in the primitive Earth. Some of them may have actually accumulated from infalling material. I will address this issue right away. So I think that now we accept the idea that there was indeed a synthesis and accumulation of organic compounds in the primitive environment. There are several lines of evidence that I will discuss with you, recognizing, if not at the origins of life, at least during very early processes, very early biological stages, what we call now the RNA world. And many of us will agree, actually a significant majority will agree, that in this, from this RNA world is that protein synthesis developed, the DNA appeared, and these were primitive cells. So keep in mind that I'm always referring to, if not explicitly, at least implicitly, to the presence of lipids, of membranes, and a very complex set of molecules held together by some kind of compartment. So what I will try to do now is to convince you that this scheme that recognizes 3 major stages is indeed valid, and that we have lines of evidence supporting it. So let me start with the idea of the synthesis and accumulation of organic compounds. Both in the traditional operating perspective and in the current assessment of the theory, we accept that there was a synthesis of a non-biological synthesis, what we call a prebiotic synthesis of organic molecules that were of biochemical significance. And in fact, we now recognize that most likely those molecules appear actually prior to the formation of the Earth itself. And the evidence comes from astronomers. Now, it's impossible to demonstrate that the scheme that I showed to you is correct. Nobody was there when life appeared. None of us has direct proof that this is the way that life appeared. But I think one should appeal philosophically to what Darwin in fact used, which is consilience, which is the coming together of evidence from different fields. And I think it's fair to say that it's impossible to demonstrate that this is the evolutionary pathway that led to the origin and early evolution of life. But I think we have enough evidence to agree that all the available evidence from widely different scientific fields is consistent with the possibility that it happened this way. And this is precisely what I will try to show you and convince you in the most critical manner I'm able to do so. Now, as I already mentioned, both the traditional perspective and the current ideas actually suggest that there was a synthesis of organic compounds prior to the appearance of the Earth. Many of us are convinced that they took prior to the formation of, prior to the appearance of life. Many of us are convinced that they took place prior to the formation of the Earth itself, and the evidence comes from the astronomers. We know that in the late stages of evolution of solar-type stars, we have what we call red giants. We can actually see simple molecules like CN, CC, OH, and so on in the outer surface of the atmosphere of these stars. We know that these molecules are expelled into the interstellar environment. We know that these, because we actually see them, we know that these organic molecules undergo further changes in the interstellar clouds, where even though there are high levels of UV and X-ray radiation, nevertheless, the clouds are protected by all the dust, the graphite, and the carbon grains that are there. And we can actually detect a huge array of more than 150 different molecular species in the interstellar medium. We actually see the condensation of these interstellar clouds into planetary systems. This is no longer a hypothesis. This is something for which we have excellent theoretical and observational evidence. And the idea is that the material that was circling around the primitive Earth, that we see circling around stars in formation, actually led to the formation of larger bodies like asteroids or comet-like structures that begin coming together, colliding, and from this process, the planets were formed. Now, we can not only hypothesize that this happened in the solar system, but we have direct proof that it happened in our own neighborhood, as seen in the next slide. This is a picture taken from the moon. You can see here the Earth seen from the moon, from a satellite in orbit around the moon. And you actually see the huge amount of craters that are still well preserved in a 4.10 to the 9 years old moon surface. The moon surface is very highly conserved, very highly preserved. It looks like the Earth was 4 billion years ago, for a very simple reason. The moon is smaller than the Earth. The ratio of surface to volume is greater than, hence the moon cooled down very rapidly. So we have a frozen evidence of how the Earth may have looked. And most of these craters are in fact about 4 billion years old. And because the moon is so close to the Earth, we can, without any doubt, we can be sure that actually if the moon was subject to this very intense flux of smaller bodies falling upon its surface, clearly the intensity of equivalent collisions must have been greater over the Earth. And consider that the Earth is more massive, so we would have attracted much more bodies than the primitive moon did. Now, what kind of bodies actually fell upon the Earth? We actually have very good evidence, as seen in the next slide. In some cases, we can reconstruct the orbits of the bodies that fall upon the Earth. Here you see you don't see it, but believe me, the position of the Sun is here. We have the orbits of Mercury, of Venus, the Earth, Mars, part of the orbit of Jupiter here, and we see part of the zone where the asteroids are, the asteroid belt. And in some cases, using a methodology that was developed when Gauss, the mathematician Carl Gauss, was only 16 years old, which always humbles me and should humble all of us, we can actually calculate, we can determine without any big problem the exact orbit of infalling bodies. In this case, of for meteorites, the Pribram, the Innisfree, the Low City, and the Peckskin. And if you look at the perihelion of these, if you look at the focus of these elliptical orbits, you see that they all fall well between the zone of the asteroids. Now, it's a zone that we know extremely well. It's a zone for which we have superb indirect chemical analysis. This following picture, which you cannot see clearly, unfortunately, is just a photo mosaic of several bodies that come from the asteroid region. This one, in fact, is Phobos, one of the moons of Mars. That is a captured asteroid. So here we have the evidence of these primitive collisions. And the scheme that we all accept is that many of the meteorites that fall upon the Earth right now, as seen in the next slide, actually are due to the collisions of the intersection of the orbits of the asteroids. And you can see in the next slide how this asteroid will collide. The fragments will be sent in all directions. Some of them will fall towards the inner part of the solar system. And this is what we can later see on as meteorites, as seen in the following picture. This picture, it doesn't show here. It's a gift from my friend Dave Deemer. It's an extraordinary picture of infalling meteorite during daylight. And just because of the incandescence, just because of the friction, you can actually see the light it is emitting. And that is exactly what happened in 1969 in Australia, near the town of Murchison. At 10:55, the Australians were very eagerly awaiting for the pubs to be open. They opened at 11, and all of a sudden at 10:55, a meteorite was seen on the sky. The meteorite broke in many parts, and you can see in the following scheme. Well, here's the town of Murchison, and well, you don't see it, but the meteorite pieces fell over this zone. They were very quickly collected. Mostly of them ended up in the United States, and we can analyze them without any problem. I like to underline the fact that the meteorite fell on the Earth in 1969 because by then, NASA had already installed the laboratories for the analysis of the lunar samples. And we could use them without any problem to actually analyze, with diminishing the risk of contamination, the meteorites that were coming, or the pieces of the Murchison or any other meteorite that we can collect. Now, it sounds very impressive to say that we can analyze meteorites, but in fact, if we can see the next slide, it's something that can be easily done. We have the piece of the meteorite. I'm sure that few of you can see that the outer surface of this piece of the Murchison is darker, it's much more altered, but we can actually take samples from the inside of the meteorite. And remember, this particular meteorite is sort of a stone. It's what we call an stony meteorite, a carbonaceous chondrite that happens to be rich in organic molecules. Because only the surface of the meteorite is altered, we can actually analyze without any problems the inside. And it's only the surface has been changed because rocks are very poor heat conductors. If it were a metallic meteorite, all the meteorite will be heated all over its body and then everything will be altered. Not in this case. In this case, we can actually take just a piece, a sample from the inside of the meteorite and analyze it, being sure that it has not been altered by the harsh conditions it encountered when it actually collided upon the Earth. Now, whenever we do this kind of analysis, what we found is that, first of all, the meteorite is 4.16 to the 9 years old. In other words, that when we studied the Murchison and other meteorites, we have a sample of the chemistry of the primitive solar system. In a sense, we have a fossil of the chemical events that were taking place in the primitive solar system. And secondly, although analyzing a meteorite appears to be very complicated, you appeal to exactly the same methodology, exactly the same techniques that you will have if you go to the physician and want to have your levels of cholesterol analyzed, of your levels of sugar in your urine. It's exactly the same methodology, and it's simply showing us the power of chemistry when applied to cosmic bodies. When this is done, and specifically with the Murchison meteorite, it's fascinating that we see a significant fraction of organic compounds of something that we call kerogen. Kerogen is a broad name for a polymer of undescribed nature. It's very difficult to analyze it. Once I tried to do so, and it's really, it's a tar-like material, very much like chapopote, as we call it in Mexico. It's very complicated. We really have no idea if it's just a generic name or refers to a set of reduced set of molecules. But what is fascinating is that there is a fraction of soluble organic materials, and this includes carboxylic acids. It includes all the decarboxylic acids that we see in the Krebs cycle. We see about 90 amino acids, we see amides, we see purines, we see remnants of sugars, and so on. So clearly, what the meteorite is telling us is that 4.6 10 to the 9 years ago in the solar system, we had chemical synthesis going on that were producing not all kinds of chemicals, not all kinds of molecules, but some of the molecules that we associate with biological activity. As seen in the next slide, we don't have a piece of the primitive Earth to analyze directly. We don't have any remnants of the primitive atmosphere, but you know that in 1953, Stanley Miller, who was then working on his PhD thesis under the supervision of Harold Urey at the University of Chicago, decided to model the chemistry of the primitive atmosphere. He used a system in which there was a mixture of methane, ammonia, hydrogen, and water vapor that was coming from the lower flask. And in only a few hours, Stanley Miller was able to show that you could synthesize a number of organic compounds without any problem. So what he was really trying to do was to simulate under laboratory conditions the possible primitive atmosphere of the Earth, and he was able to show without any doubt that it's very easy to synthesize organic compounds following the ideas of Oparin, following the model of an atmosphere developed by Urey, and so on. One thing that I like about this picture is that it's a composite picture. This one comes from my friend Bill Schopp at UCLA, but what I like about this picture is that it shows how our standards have changed. You can see that the upper flask was covered with asbestos, amiant, which is something that no one in his right mind will use today. And what is also very significant is how the dressing code of the graduate students have changed, because at the time, the people had to go with black shirt, ties, and cufflinks into the lab, and now you see only in some university authorities and a few old-fashioned professors like me. But so some things have changed, but not necessarily for the better. In any case, what Stanley Miller did was to take a sample of the compounds that he had synthesized. He then performed a very simple paper chromatography analysis, and what he was actually able to demonstrate is that you could synthesize glycine, aspartic acid, alpha-alanine, beta-alanine, and alpha-amino and butyric acid, and a few additional compounds. I was a very close friend of Stanley Miller. I was lucky to be very, very close to him, to work with him. We published a number of papers, and then he very kindly gave me a sample of the original experiment that he had kept. And so we reanalyzed them together with my friend Jeff Beda at UCSD, at University of California, San Diego. And just by using the contemporary techniques, it's absolutely amazing the amount of information you can get. Here is the outcome of one of such analyses. You can see here a list of the different amino acids. Stanley Miller had reported 5 of them. We actually found 22 different compounds, some amino acids that had never been reported. I was particularly enticed by the presence of ornithine. I will come to this later. But one thing that is immediately clear is that it's not that Stanley Miller failed in doing the proper analysis, it's just that our current methodologies are much more refined, and we can actually have very, very good insights on the chemistry of the simulations of the primitive Earth. One thing that has to be said also is that sometimes the people have the simplistic assumption that all the compounds necessarily for the origins of life simply were formed in the primitive earth, and that cells appear from that mixture, and that was it. Let me show you how the case is not really so simple, and I will put two examples of that. Here we have valine, which is of course a proteinic amino acid, but you can see also that isovaline and norvaline, which are isomers of of valine, are formed. In fact, isovaline is formed much more abundantly than valine. So that means that if these experiments are actually proper simulations of the primitive environment, we have to recognize that there was a period of selection, chemical selection due to physical stability, to chemical reactivity, whatever, that actually selected some of the molecules that we see in living entities today from this very complex mixture that must have been present in the primitive earth. Now, there are other ways of demonstrating this, as shown in the next slide, namely that how good are our experiments as compared to what the meteorites are telling us. And well, we had the chance to analyze the Murchison meteorite, a sample of the Murchison meteorite, again in Jeff Veda's lab. And here you can see we decided to go for the aliphatic amino acids. For those of you that are initiated in the complexities of the chemical language, you know that aliphatic means that amino acid like glutamic, alanine, aspartic, beta-alanine, and so on. And here you have in blue the amino acids that we detected, the aliphatic amino acid that we found in the Murchison meteorite. This was not the first report, we simply redid the analysis. But here you have the comparisons with the amino acids that were synthesized under a repetition of Stanley Muir experiment. And you see a superb correlation between the blue bars and the black bars, showing that in fact our simulations are good representations of the chemistry of the primitive solar system and perhaps of the chemistry of the primitive Earth. Now, having said that, in some cases the situation is not so simple. So let me underline that if we look at the list of the entire amino acids, we actually see over 90 different amino acids. I really don't care if you cannot see this picture, because even if you have the table in your hand, it's very difficult to analyze. In some cases, we actually detect the lines of amino acids, but we cannot identify them because we don't have the standards, we don't have the chemical standards. But the chemical complexity of a meteorite is extraordinary, of a carbonaceous chondrite can be extraordinary. So clearly, one should not jump to quick conclusions about how complex was the primitive soup, how complex was the prebiotic broth, considering how efficient the synthesis was in the case of the parent body of the meteorites. What is very surprising, as you can see in the following slide, is the fact that amino acids are abundant in chondritic meteorites and are readily formed in model prebiotic experiments like those of the Mueller-Urey experiment. But this is not true of arginine and lysine, which are basic amino acids. A week ago, a friend of mine, John Sutherland, reported the possible prebiotic synthesis of arginine, but I will come to that later. And we do not see histidine, which is, of course, the only amino acid, the only biological amino acid that we know today has the imidazole group, which is a highly reactive group. So what we really have here is a demonstration that not all the molecules present in meteorites or synthesized in a prebiotic experiment made it to biology. Not all the compounds that we see in biology were formed or were available in the prebiotic environment. Now, one thing that I like to point out, and it's especially relevant for some of the discussions I think that we will have later on, is that 1952 was an extraordinary year by all accounts. It was the year in which Stanley Mueller demonstrated that you could synthesize organic compounds under possible primitive air conditions. It's also the year in which Watson and Crick proposed the double helix model. In fact, the two papers were published in a journal difference, one in Science, the other one in Nature, and they were published with only 3 weeks of difference in time lag. What is absolutely fascinating is that in many ways, the publication of the Watson and Crick double helix models epitomizes a process of modularization of life sciences. And we know what happened. All of a sudden, the physicists were displaced from this key role that they had socially, politically, economically, scientifically, as the representative of what a scientist should be. It also happened that biology underwent a process of extremely reductionism that led to major discoveries about what genes are and what they are, how they are expressed and so on. But it's equally true that some parts of biology suffer tremendously. It's true not only for the people doing natural history, that all of a sudden were displaced from this key role of what biology is, and everybody was concentrating on the magic of double helix. People start to work to forget what the biology traits of an organism are just by concentrating on DNA. And at the same time, clearly the origin of life necessarily to be validated under this new era that was inaugurated by the publication of the double DNA helix model. Clearly the origin of life needed to be inserted in this context. And indeed that insertion took place a few years later when my friend John Oro, and then at the University of Houston, was able to synthesize adenine from a very simple solution of hydrogen cyanide. Hydrogen cyanide is extremely poisonous. All of you know of all those characters in novels and so on. Their spy novels are fascinating, very quickly killed, very efficiently killed by the ingestion of hydrogen cyanide. It's that typical almond aroma, but in pure solution, hydrogen cyanide can undergo dimerization, trimerization, tetramerization, and John Oro was able to demonstrate that it reacted with formamidine, which is also a derivative of hydrogen cyanide. It will form the imidazole group, and then with more reaction, again with formamidine, it will form the imidazole group. And then a few years later, Jim Ferris and Leslie Orgel, they were actually working in the patio of the Salk Institute, the patio designed by Barragan, the famous Mexican architect, and they had a vial with water with some hydrogen cyanide solution in which they already had the tetramer, the aminomalonitrile, which is a highly reactive molecule, and they simply exposed that vial to the sunlight, and spontaneously the molecule underwent a photoisomerization, and spontaneously it underwent a second photoisomerization to give the imidazole group, and then the an additional hydrogen cyanide molecule to give adenine. So it's very easy to synthesize under possible prebiotic conditions in a very simple way, a solution of hydrogen cyanide with some light. It's very easy to synthesize one of the components of DNA, one of the components of RNA, one of the molecules that play a key role in bioenergetics. So this has strongly validated the field of the origins of life for a long time. Now there was a link between origins of life and molecular biology. And a few years ago, together with Stanley Miller and John Oro, we decided to see if other bases could be formed from the imidazole group. And indeed, we were able to show that once you have the imidazole group, which is a highly reactive molecule, we will have not only adenine, but also guanine. And then, of course, you can, at the same time, by an desamination, an oxidative deamination, you could have xanthine from guanine and hypoxanthine from adenine. So you can have two biological bases from a very simple molecule under possible prebiotic conditions. And then, because we were very interested in seeing if additional bases could be formed, we actually performed a simple experiment shown in the next slide, in which we actually, well, I'm just sorry it doesn't show, but believe me, we took a solution of a derivative of cyanide. We heat it up to 80 centigrades, which is a very reasonable geological temperature. And we saw, of course, the peak of adenine. We saw, of course, the peak of guanine, but we also saw the peak of 2,6-deaminopurine. 2,6-deaminopurine is a purine that we don't see in RNA, we don't see in DNA, but it's a molecule, as shown in the next slide, that without any problem can actually form hydrogen bonds with uracil. So this raises the possibility that under primitive Earth conditions, during the earliest stages of biological or chemical evolution, there were additional bases forming complementary base pairs. And this possibility has been strongly supported by more recent work summarized in a recent paper by Andros Rios and Ishak Tor, in which they showed the huge number of purines and pyrimidines that you can synthesize prebiotically in experiments just like the ones I have described, or found in meteorites. So it's perfectly feasible that the primitive genetic material was endowed with a greater diversity of functional base pairing forming bases, both purines and pyrimidines. I'm not, I think it's reasonable to say when you look at all the experiments that had been done, that although we lack a precise description of the conditions of the primitive Earth, I think it's reasonable to say that the primitive Earth was an environment in which, thanks to the geological activity, to electric discharges, to radioactive decay, to infalling material, comets, meteorites, and so on, in an atmosphere that was most likely rich in CO2 but had some methane, that also had some ammonia, hydrogen sulfide, and so on. There was the formation of amino acids, nucleobases, sugar, lipids, and oligomers of biochemical molecules. There is a trend, a tendency, I would say, that I think is slightly unfortunate among some of my colleagues, some, not all, of my colleagues working on the origins of life, because they have a fixation for one particular source of energy or one particular source of organic compounds. The people will say the most important were the compounds formed in volcanoes. Others will say, no, the most important ones are the ones that came in meteorites or comets. Other people say, no, the most significant ones were the reactions that took place in hydrothermal vents. But I think this is the typical Anglo-Saxon discussion of a typical Anglo-Saxon gastronomical discussion. Why is the reason why the food is so good in the Mediterranean, in India, in China, or in Mexico? That's because we eat everything. If it works, it's edible. You never despise a source of organic compounds. And I think that we better have a very eclectic vision of how to cook the primitive Earth, having sources of organic compounds from different environments under different conditions, and hence be able to form a much more rich, complicated soup than with just one simple gastronomy, let us put it that way. I will not refer to any additional culture or society in this way. So I think one should really be very eclectic in recognizing the complexities of the primitive environment. I'm not going to go into the details, although I love them. I think I'm a closet chemist more than anything else. I will not go into the details of the chemical processes that may have taken place on the primitive Earth, but I think it's fair to say that all the experiments and all the analysis that we do demonstrate that in the primitive Earth there was a wide array of organic compounds of biochemical significance. Not necessarily all of them found their way into extant living entities. There were many inorganic and organic catalysts. The people make a lot of the presence of catalysts, but remember that a proline, an amino acid, can be a very efficient catalyst, that clays can be efficient catalysts, that metallic ions can be catalysts, depending on the conditions that we set up. There were clearly purines and pyrimidines, which means the potential for template-directed polymerization, and there were membrane-forming compounds. Not necessarily as complex as the phospholipids, in fact, quite unlikely the phospholipids that we see today, but there were possible a wide range of molecules that could form quite easily membranes, because being hydrophilic, having a hydrophilic component, a hydrophobic component, you can easily form double membranes, you can easily form micelles, you can easily form liposomes. So the chemical complexity of the primitive Earth is such that we see a chemical composition somehow equivalent to the one we see today. We could see catalysts that we associate normally with metabolism. We see the potential for replication that we associate with reproduction, and we see membrane-forming compounds that we associate with entities. So some of the basic traits of life were already there. Now the question is, how do we go from that that mixture, that very complex mixture of organic molecules in the primitive Earth, to something that we recognize as one of the basic traits of living entities. Excuse me for the simplicity of this scheme, but all of us recognize it without any problem. And you know that for a long time, the issue was whether DNA came before proteins, or if proteins came before DNA. And this was the source of a tremendous level of discussion that went beyond the scientific analysis. It was actually, you could see how the people were divided even in political camps. Some of my most leftist colleagues accepted the idea that proteins came before DNA, and they were basing their suggestion on the misreading, I think, of Engels, which can be very boring, and many people did not realize that Engels had taken those ideas from Haeckel. Some of my most conservative colleagues were actually using these horrendous anthropocentric metaphors to say, well, DNA is like the general of the cell, or is like the manager of the biochemical activity, and so on. And the debate was in fact quite intense and went beyond the limits of labs, of conferences, of libraries, and so on. In fact, what most likely came both before proteins and DNA, as we know now today, is precisely RNA. And if I can have the next slide, the question is how, well, you don't see it, but in any case, the idea that RNA came before both proteins and DNA has a long historical it can be, the history of this idea can be traced back to the extraordinary insights of people in the 1960s, 1950s who were recognizing the significance of coenzymes that have a ribonucleotide component, of people that recognize that indeed RNA can store genetic information, that RNA can take very complex three-dimensional shapes. But nowadays, I think that most of us recognize that there is very little doubt that biological catalysis has to be understood not only in terms of the activity of enzymes, but also in the activity of ribozymes. I have to tell you very quickly a short anecdote that I actually witnessed. I was doing some experiments in the laboratory of my friend John O'Rourke in Houston when the papers on the catalytic activity of RNA were published. So I quickly ran to his office and said, John, look, John, Juan, Juan, whatever. I told him, look, yeah, well, he was Catalonian, so he insisted on being called Juan, but Juan is a female name in the United States, so he was not enticed by those confusions. But I went to see him and say, look, John, here we have the evidence that there is catalytic activity of RNA. And John looked at the paper in a very dismissive way and say, yeah, but it's only hydrolysis of phosphodiester bonds. Anyone can do a hydrolysis of phosphodiester backbone. Well, nowadays the situation has changed dramatically. I'm very fond of this table that was prepared by my friend, my colleague, my student, Ricardo Hernandez, who's sitting over there with his girlfriend. It's a fascinating table because here you see in every line the different classification of enzymes according to the International Union of Biochemistry. So we know that enzymes can be classified as enzyme commission number 1, oxidoreductases, enzyme commission 2, transferases, enzyme commission 3, hydrolases, enzyme commission 4, lyases, enzyme commission 5, isomerases, and enzyme commission 6, ligases or synthetases. And here you have examples of the enzymes that actually perform the activity described by their name. But in the last column, Ricardo very carefully looked at all the available reports in the literature, and here you can see how we also have RNA, catalytic RNA ribozymes that actually act as oxidoreductases, as transferases, as hydrolases, as lyases, as isomerases, and ligases. So all the activities that we associate in enzymes can also be found in catalytic RNA. True, some of these activities have been actually selected under laboratory conditions, but the fact is that we have no doubt that RNA is in fact an insignificant biological catalyst in extant biological systems. Now, one thing that I think is fair to say, and shown in the next slide, is that, of course, we know also that RNA is endowed with additional properties. For instance, RNA can fold upon itself, and we can have the riboswitches, which means that the RNA molecule by itself can control the expression of the genes it is encoding, which is not a minor trait. And in fact, as we can see in the next slide, I think that the catalytic, regulatory, and structural properties of RNA molecules, combined with their ubiquity in cellular processes, are consistent with the proposal that they play a key role in the early evolution and perhaps in the origins of life itself. And let me give you some examples of how those situations in the next slide. We know that if we want to have RNA, we need to polymerize ribonucleotides. Ribonucleotides are synthesized universally by few common steps. That means that it's a monophyletic trait that clearly appears very early on life. But once we have RNA, these RNA molecules can be, as seen in the next slide, can be seen as coding RNA, as in the case of viruses, as in the case of RNA viruses, of course, as in the case of messenger RNA. We can have regulatory RNA, as seen in the next slide. We can have large non-coding RNAs, as we can see in the ribosome, as in the primer, as in transfer RNA. And in fact, we also, if we want to have DNA, don't forget that we need to form the deoxyribonucleotides, we need to reduce the ribonucleotides. This is a universal biochemical trait. It may be that the deoxyribose was formed under primitive conditions, but biologically, once you have natural selection in a biological system. If you want to have DNA, you necessarily need ribonucleotides. So I think this is a very strong argument based on biochemistry, based on what living systems are telling us today, is that if we want to have DNA, you need prior the synthesis of RNA. And one thing that we are concentrated in my lab, in my group, with the students, colleagues like Arturo and others that are around here, is to try to look at the other derivatives of ribonucleotides that for a long time have been sort of the ugly duckling, if you wish, in our description of the RNA world. And these refer, in fact, as in the next slide, to coenzymes, to alarmones, and to histidine. So let me go first to coenzymes. They are, in fact, we recognize them as possible remnants or providing evidence of the role of RNA in early biological evolution, thanks to the work of extraordinary people like Belozersky in the Soviet Union, Philip Handler in the United States, a major biochemist who actually became president of the National Academy of Sciences, by people like Eakin, who was working independently at Austin at the University of Texas. And independently, and later on, Leslie Orgel, they recognized that, first of all, many enzymes require to perform their catalytic activity, require the presence of coenzymes, and many of the coenzymes, about 50% or so, are actually ribonucleotide derivatives. Here you have different coenzymes. I'm not going to go into the details, but you can see actually how they are, in fact, derivatives of adenine. So this by itself is telling us that, although one point that has to be emphasized, a nucleobase, adenine 1 in purine by itself, is not chemically reactive. They just collapse and they are basically hydrophobic. Well, they are not basically hydrophobic, they are hydrophobic, and they simply separate from the aqueous environment, and that's it. But if we have the base combined with the phosphate, with the sugar, and with other residues, then we have them as coenzymes, and they are essential for biological catalysis in extant living system. The other issue that we are concentrating in the lab is, and this is a work I did together with my friend Luis Zelaya and my friend Arturo Becerra, as shown in the next slide, are alarmones. Alarmones are, you are all aware of the role of cyclic AMP. Alarmones are biochemical signals, second messengers, if you wish, that signal a stress condition, either outside stress or inside stress, like the lack of water, like the lack of nutrients, like high UV densities, and so on. And one thing that Luis, Arturo, and myself realized very quickly is that if you look at the alarmones, none of the alarmones are deoxyribonucleotides. All of the alarmones that we are aware of are, in fact, molecules that are either ribonucleotides or derivative of ribonucleotides. And one thing that, this is part of the thesis of Ricardo Hernandez, again, who's over there, still with his same girlfriend. One of the things that we actually realized is that we can, without any problem, come out with very simplistic ribosome-mediated catalytic reactions to synthesize all of these alarmones without any problem. There is a practical issue that we are now investigating, which is not a minor one. Of course, we know that when you have a patient in critical conditions, you have a patient in the intensive care unit, the bacteria, the microbiota associated with him, put him under a stress condition. Every patient in a hospital is under a stress condition, if not because of the illness, at least because of the medical bill that he or she will have to pay. But one thing that is absolutely true also is that also the bacteria, the microbiota infecting the patient, are in fact under stress conditions, because we are showering them with lots of antibiotics and so on. And Ricardo Hernandez discovered, and this is an issue we are now discussing with Samuel Ponce de Leon, one of the foremost infectologists in Mexico, the place where we found most alarmones, more abundantly expressed alarmones, is precisely in the intensive care unit of hospitals. So this is something that may have actually a practical application, but the fact is that all of them are ribonucleotide or ribonucleotide derivatives, is telling us something about how very early in evolution derivatives of ribonucleotides or RNA actually began to play a very important functional role. And finally, the point that I'm very enticed with now is, as seen in the next slide, is histidine. Histidine is a typical amino acid. You here see the carboxyl group, the amino group. But what is very peculiar of histidine is the fact that histidine has an imidazole group. And in 1972, Harold White proposed that, in fact, histidine should be seen as a remnant of a ribonucleotide. And why? Because we know that it's the only amino acid that has the imidazole group, such as the one we see in adenine, for instance. And furthermore, because we know that histidine is a major catalytic amino acid. The amino acid, thanks to this hydrogen, it can play a key role in acid-base catalysis. And just a week ago, one of the students of the lab, Betunes Betabeles, Alberto Vasquez, actually presented his undergraduate thesis when we actually analyzed in full detail the biochemical pathways leading to histidine. And what is fascinating about histidine from an evolutionary perspective is the following. If we have proteins, we can have enzymes, and we know that enzymes, as seen in the next slide, must be very ancient in biology. We do not know, and this has to be emphasized over and over again, we do not know how proteins first evolved, but clearly their active sites must be one of the oldest components of enzymes. I think that's a very general conclusion on which everybody will agree. Now the question is, what do they tell us about their catalytic components? And there was a fascinating paper that was published, and we have repeated the analysis, and it's of course quite correct. It was published by an English group. And once again, you have here the different classes in this line, EC1, EC2, EC3, EC4, EC5, EC6. You have the different classes of enzymes according to the International Union of Biochemistry. And here you have the most abundant amino acids in their catalytic site. And it's perfectly clear that histidine is by far the most overrepresented amino acid in the active site of enzymes. So what biology is telling us is that if you want to understand the origin of biological catalysis, if you want to understand the origin of protein catalysis, if you want to understand the origin of enzymes, you really need to understand the origin of histidine and how did it came to be located in that key position. So let me summarize my love for ribonucleotides by saying that although they have been despised in our descriptions of the RNA world, they are monomers of RNA, they are energy-carrying compounds, ATP, GTP, UTP, we are all familiar with those things. They are precursors of the exoribonucleotides, they are coenzymes or precursors of coenzymes, they are alarmons or precursors of alarmons, and they are precursors of histidine, quite clearly. So I think it's fair to say that there are many discussions on what was the RNA world. We don't have a very good definition on what the RNA world is. There are many idiosyncratic definitions of the RNA world, but in spite of the existence of many definitions of the RNA world, including several that are contradictory among them, one could say that it's an early, perhaps primordial, stage during which RNA molecules play a much more conspicuous role in heredity and metabolism. And the role of ribonucleotides and modified ribonucleotides as components of the RNA world should also be considered. So we are proposing that if you want to have RNA, yes, but you must also have ribonucleotides and all these derivatives as well. So even if people do not accept, because there are some skeptics, there are some people that will say, no, there was never an RNA world, it played no role in early biological evolution, it's impossible to synthesize under prebiotic conditions and so on. You can listen to the talk by my friend Ada Jonah to be convinced otherwise right away, but it's perfectly clear that even if you don't accept the fact that RNA may have been existed on the primitive Earth, even if this possibility is not accepted, the origin and the manifold roles of RNA in extant biology need to be addressed without any doubt. Now, why do some of us believe that the RNA world hypothesis is very robust? Because we can have RNA molecules that act as replicases, that under laboratory conditions can replicate other molecules of RNA. We can have a small ribozymes of no more than 200 ribonucleotides that actually catalyze metabolic reactions. And of course, the best example of the catalytic abilities, the catalytic properties of RNA is the ribosome. But of course, having Ada Jonah here, I will not dare to say anything about that, except that it's a very clear indication of the key role that RNA molecules play very early in biological systems. So let me share with you what I think are some important conclusions. First of all, extra-natural ribozymes, not the ones that we synthesize under laboratory conditions, the ones we see in cells, acting today in cells, are mostly involved in RNA processing and in protein biosynthesis. In fact, the ribosome is a ribozyme. This is a wonderful mantra that you could repeat over and over again, and it's going to be absolutely true. Secondly, both the catalytic and the structural properties of the ribosome strongly suggest that protein synthesis evolved in an RNA world. But again, Ada Jonat will address this issue. This implies that the origin of the genetic code and the origin of life are not necessarily synonymous. I know that there are opposing views here, but I'm absolutely convinced that once you have something that's being encoded by biology, even in the absence of triplets, then you don't necessarily need the genetic code. And finally, since DNA replication is catalyzed solely by proteins, we know of biologically, there's no role of ribozymes in the replication of DNA. It appears that it is the outcome of biochemical evolution and not of abiotic processes. In other words, one who has to distinguish very clearly the stages of chemical synthesis, pre-biological evolution, the RNA world, and what we will recognize as a living system quite comparable to the ones that we see today. So, I think it's fascinating to look at the history behind the RNA world hypothesis. People profited from the extraordinary chemical expertise of especially the European, the German school in the 19th century. This provided evidence that you could synthesize organic compounds under laboratory conditions, and the borders separating the non-living from the living were erased. It was a major philosophical achievement. At the start of the 20th century, the people did not know how photosynthesis took place, but it led to a number of models of photosynthesis that in turn reinforced the previous discoveries of the extraordinary achievements of organic chemistry. Then in the period between 1924, 1930, more 1929, independently, Oparin, Haldane, Liebman, Harvey, and others proposed the idea of heterotrophic origins of life. I think that Oparin contribution is the most significant one because in fact he proposed a research program that is still valid today. Then we had in 1953 the Miller experiment that really validated in the eyes of many the significance of the origins of life studies. We had the synthesis of adenine by John O'Rourke, which connected it to molecular biology, and then with this amazing, extraordinary period of realizing that RNA is an essential molecule in extant living beings and very likely also present in early biological systems. So this raises a fascinating issue. A key point in the study of the origin and very early evolution of life is how did the RNA world came into being? And here I have to put big question marks because we really do not know. We recognize the role of RNA. We are perfectly happy with the way it helps us to understand the origin of protein biosynthesis and so on, but we really do not understand how RNA first evolved and first accumulated on the primitive Earth. There is a very recent paper, a fascinating paper that I strongly encourage you to read. It was published in Nature Chemistry by John Sutherland, the English group led by John Sutherland. And of course, you cannot see nothing here. Even if the lights were off, you will still see nothing because it's a very complicated scheme. But let me very simply say that what's fascinating is that here you have very simple precursors: hydrogen cyanide, water, formaldehyde, other aldehydes present in the primitive Earth. And John and his group have come up with experimental demonstration that you can synthesize the precursors of lipids, the precursors of nucleotides, and the precursors of a number of amino acids, including arginine that I already mentioned, that can be synthesized commonly from the same precursors almost at the same time. I think it's a fascinating demonstration of a principle, because if one reads with a very critical eye the paper, what it turns out is that, first of all, there are some chemical issues that have to do with this extraordinary paper. The conditions are highly constrained. You really have to have very specific pH values. You have to be changing the amount of water and so on all along the experiments. I think it's unlikely that there was as much as iron as he was assuming on those experiments. Iron would have very quickly reacted with hydrogen sulfide present on the primitive Earth and will form sulfide, iron sulfides. Hydrogen reacts with ammonia and aldehydes in a much more efficient way than it is assumed on the paper. The scheme is based on a number of unreasonable reaction concentrations and a number of ad hoc assumptions like different values of pH and different temperatures at different stages of the experiments. But what is absolutely fascinating is the demonstration of a principle. From very simple molecules, you can synthesize at the same time all the components or the precursors of component of extant living system. And I think that's an essential point that should be kept in mind. The problem, as seen in the next slide, is that if we want to have RNA, we need to have ribose. And if we want to have ribose under primitive conditions, we are in a big problem. First of all, because sugars are chemically unstable. That's the reason people get drunk with cocktails. Sugars are extremely unstable chemically, and under primitive Earth conditions, they will be absolutely true, even more. In a prebiotic mixture, like one of these Miller experiments I have been describing to you, ribose could exist, first of all, as an open chain aldehyde, or it could exist as the alpha and beta pyranose and furanose form. The ribose that we see in RNA is a ribofuranose, but it will be in equilibrium with the other forms under prebiotic conditions, in water conditions. And then we will have both the L form and the D form of sugar whenever we try to do a chemical synthesis. And it's not clear at all why life became so asymmetrical in chemical terms. It's something that, again, needs to be kept in mind. We don't have good phosphorylation chemistry for the primitive Earth. There are a number of schemes. One of them was published 2 days ago in Origins of Life and Evolution of the Biosphere by Matthew Pasek. It's a good scheme showing that you can get some phosphorus that can be oxidized from a meteorite and so on, but still the people are not so convinced, and there is no obvious prebiotic oligomerization mechanism. Of course, you can have a solution of nucleotides, you can heat them and dry them, you can heat them and dry them, you can add water and so on, and in these wet and dry cycles, you will have a polymer, but it's going to be a very small polymer, and we will have the 3' 5' bonds, typical of nucleic acids, but you also have lots of 2' 5' bonds, which are absolutely untypical of nucleic acid. So it's not clear that we are in a position to really say that we understand how this has actually been overcome under prebiotic conditions. So I think that currently, and I will encourage you to read a fascinating paper published by my friend Ram Krishnamurti in the most recent issue of the Israeli Journal of Chemistry. It's a volume honoring the contributions of Albert Eschen-Moser, a towering giant in the field of chemistry. And what Ram is saying is, okay, under prebiotic conditions, we will have hydrogen cyanide, formaldehyde, formate, formaldehyde, a number of aldehydes, ammonia, inorganics, the components of ingredients, sources of phosphorus is still largely unknown, minerals, energy, and so on, and so on, and so on. And there are two possible, right now there are two schools trying to understand how RNA originated. One school, which is very, very powerful, is assuming that there were, of course, the synthesis of the ribonucleosides of the bases, the RNA building blocks, and we will have RNA. The other one is, let us assume that in fact RNA was preceded by what we call the pre-RNA world, such as the peptide nucleic acids and so on. And I think it's not the key central issue on the origins of life, but I think it's a very important point that we have to take into account. So in summary, in trying to end this lecture, which are really two lectures combined of one speaker who's absent and myself, I think it's fair to say that it's easy to form a primitive soup. One can have a primitive soup either by doing chemical simulations, by having contributions from outer space and so on. We have no problem with the primitive soup. We need to investigate the actual stability of compounds, the synthesis of some, preferent synthesis of some compounds over others and so on. But it's really very easy to synthesize organic compounds and have a primitive soup. One can look at databases and this extraordinary, unexpected, rapid development of bioinformatics, more efficient computers, an enormous amount of data and so on, actually allows us to have very reasonable ideas about the early phylogeny of life, the early evolution. It's clear that DNA is a latecomer in evolution. It's clear that DNA must have evolved in a system that was already endowed with protein biosynthesis. The code was already here, and all the evidence that we have strongly suggests that indeed protein biosynthesis evolved in an RNA world. But we have still the big question mark I told you from the very beginning will be present all over the lecture. We do not know how to go from the primitive soup into the RNA world. We do not know how to get ion channels. We do not know how to synthesize ATP. We do not know how to entity. It's a huge set of problems. But one of the fascinating things of science, I'm sure you will all agree with me, is that in science, it's important not only to have the answers, but also to be able to pose the proper questions. Thank you very much.
Well, we have time for a question and discussion.
Yes, we have some news in the schedule. The talk of Dr. Ada Jonat has changed to tomorrow at 19:45. So right now it's discussion and will be free until 3:30 p.m. Sorry for the inconveniences. Also, we are very happy that we have an extraordinary moderator, Dr. Rosie Redfield, from the University of British Columbia, from the Life Sciences Center. She's a professor of genetics, and her work is centered on the evolution, mechanism, and function of DNA intake in the organism Haemophilus influenzae. Redfield, nevertheless, is better known for implacable scientific rigor. In 2011, Nature magazine named her one of the 10 people who matter. This happened as a result of the professionalism with which she pointed out multiple deficiencies of a high-profile publication. Her commitment to scientific accuracy is the reason why Redfield will have the role of the discussion moderator throughout the meeting. Thank you, Dr. Redfield. And we will have a session of questions. Who is also Dr. Arturo Becerra is going to direct. Thank you.
I think I'm fine here. Thank you for those very kind words. I'm going to be picking people to ask questions, and if all else fails, I'll ask a question. But I should tell you that I'm going to be a very biased moderator in my choice of people to ask questions. I'm going to favor the audience, the students, over the speakers. And I'm going to favor, as much as I can see in the distance, I'm going to favor questions from women over questions from men, because all too often we see in the question period, it's only men asking the questions. And I'd like to sort of nudge that a little bit in the other direction. So everybody can ask questions, but if you're a male expert, don't be surprised if you're not the first person I call on.
Rosie, can I add something? In contrast to the United States, half of our tenured faculty in Mexico are females.
I noticed that this meeting is exemplary. It would not qualify for John Eisen's category of yet another mostly male meeting. This is an excellent meeting in terms of gender bias. And I'd like to keep that going in the questions as well. So, questions? Yes. You'll have to speak quite loudly because I don't think, we do have a microphone.
coffee.
Oh, and I should say, if anyone would rather ask their question in Spanish, it can be translated for them.
If you repeat the Miller experiment several times, many, many times, do you get the same relative proportions of the different components?
That's a very good question. Yes, indeed. And the number of published repetitions of the Miller experiment, not taking into account those are in high schools and so on, is well over 600 times. And in all cases, you get exactly the same results. And one point that I omitted, just because of the sake of time, is the fact that there are two experiments done, one by Michael Roth in Austria, and the other one by the same group, Jim Cleaves, John Chalmers, Jeff Beta, and myself, using CO2 instead of methane. And again, we get exactly the same results, although the proportion varies. In our case, what we did was to control the pH, because if you don't, then you go to very low pH, and at very low pH, the striker synthesis is inhibited, and then you don't get any amino acids. But if you control it by compounds that may have been present on the primitive Earth, like carbonates and so on, there you get exactly the same compounds in the same proportions. What we never get, and that's what I found fascinating, the John Sutherland experiment, we have looked very carefully, believe me, for like 2 or 3 years, we have looked for nucleobases formed at the same time as amino acids in the Miller experiment, and we don't find them. I think it has to do with the reactivity of hydrogen cyanide with ammonia, but for me it's a question that it sometimes takes my sleep away.
Okay, because I was also going to ask if there could be a very fast type of selection just based on the stability of the compounds. So you would get a broader range of components, and then this very fast action would reduce, and then you would get this very repeatable kind of experiment.
Right. It implies a certain level of chemical determinism, if you wish.
Yes.
But at the same time, you need to go into the devil is always in the details. And this is why we need to explain why, we have a good explanation actually with Claudia Alvarez that's around there. We need to understand why we don't see nor valine nor isovaline in extant proteins, although they are formed much more abundantly in all kind of prebiotic synthesis. So it's not such a straightforward conclusion as just to assume, okay, the compounds were there and living forms were formed from there.
So there might be some very stochastic
Absolutely.
processes as well.
Absolutely.
Okay.
Thank you. So there are no other molecules around or elements or anything that we do not know. I mean, every single molecule that we know is exactly the same as whatever there may be out there on the one hand, and on the other hand, the other question would be, how easy would it be to get to know a different molecule that we do not know because we're not like looking for it?
You mean in the interstellar medium or in meteorites?
I'm sorry?
In the interstellar medium or
Yes.
in meteorites? In interstellar medium, because you have lots of protons, you can have a long-lived ionized form of a compound. For instance, that was actually known since the I never read the original paper, I have to confess. But I think it was known since the 1912 or 1930. People were describing methylidine, ionized methylidine in the spectra of cometary nuclei. Methylidine is CH plus, which you never found on the Earth, of course. And then it's very easy to understand it with the interaction with the solar wind that is emitting a lot of protons. And you had an equivalent situation, in among some of the molecules that you see in the interstellar medium. But the problem is that the larger the mass, the molecular mass, the more drastic the drop in abundance. And the radio astronomers are faced with the issue of not being detecting something just because of the upper limits are so low.
Thank you very much.
Sure.
My question, well, you probably won't like my question because it involves viruses being included in the tree of life. Is there a role for the riboviruses as a link between the origin of life and the RNA world? Some giant viruses do have riboproteins.
Yes, viruses have a long historical standing in the discussion of the origins of life, because the people thought, because they are so apparently simple and apparently small, that they could be representative of the first living entities. In fact, Felix Derel, who was formally the discoverer of viruses, strongly supported that possibility. But remember that in biology, simplicity and size can be not very solid arguments. If you want a very simple comparison, you can try to, well, since we are speaking in English, you can try to compare one of Shakespeare's sonnets with a haiku from Japan. The haiku is very simple, but that doesn't mean it's primitive. It's a highly refined form of poetry that simply follow a different evolutionary path than the poetry that we see in Shakespeare in the sonnets, for instance. Shall I compare thee to a summer dream, and so on and so on. Summer wind. So what you really have there in viruses is that because all their genomes are mosaic, it's very difficult to assign an age to a virus as such. You can assign an age in some cases to some of the genes they are encoding. And the other issue is that it's feasible, but has not been demonstrated, that RNA viruses may come, some people certainly toyed with that idea, come from the RNA protein stage. I'm not so convinced that it is the case. Together with Rodrigo Jacome, who's over there, we are actually about to submit a paper showing that this is an independent demonstration that has actually been achieved by two other groups, that some components of the RNA polymerase are so ancient that you can, in fact, perhaps assume that they represent a vestige of an ancestral protein RNA polymerase. The PALM domain in particular that you see in RNA viruses is certainly the same that you see in DNA viruses and in many cells. So that may be a vestige from very ancient times, and we know that polymerases are not absolutely specific. So the transition is would be very easy to understand. But that's always in the realm of perhaps, we're not quite sure. What is very clear is that RNA viruses are a superb model of an RNA protein system, but that doesn't mean they are necessarily ancient.
Yeah, okay. I have two questions. The first one is, how do you know, or how it's known, that the craters on the moon are 4 million years old? And the second question is, what do you think of explaining life in terms of a complex system or as an emergent property?
Okay, the first question is very easy. We go there and we date the craters. I would love to be there myself. I haven't, but many astronauts have. And we have very good ages for the lunar craters. And we have no doubts about their antiquity in that sense. Now, the other question, if life as an emergent system, I think one has to beware of fashions. I think that clearly you can have complexity arising in the absence of natural selection. And let me give you a very good example. You can have, if you have lipids, you can form bilayers, liposomes, micelles, and you don't require any enzyme, and you don't require genes to form a micelle or to form a double layer. But if you compare the formation of liposomes, let us say liposomes, from archaeal lipids and bacterial lipids, you clearly see that they are forming the same kind of a structure, but the history of the components of those lipids is very different, because the biosynthesis of lipids in archaea is different from the biosynthesis of lipids in bacteria. So that is telling you that yes, you can indeed have some emerging properties in the absence of the Darwinian history behind the components. But I think that also complexity has become a buzzword that many people use a little bit too freely for my taste. I may be wrong, but I think that one should beware a little bit of fashions. And although I have no doubt that you can have, for instance, these vortices that are formed, and you have all those examples provided by Prigozhin and his group of the currents of water going up and down and so on. I think it's a far cry from those structures and what you see in actual living systems. I think they're wonderful analogies, but I think their role has to be acknowledged but should not be exaggerated.
I'm going to be an evil moderator and let myself ask a question now, because it just follows so perfectly from that question. And the question is whether the emergent property that really matters is not something that we might stick the word life onto, because we don't know what we mean by life, but rather that the emergent property is something that natural selection can act on, something that has heredity and variation, and that RNA provides us with exactly that molecule, something that's simple enough to arise by chance, but complicated enough to then evolve by natural selection into the things we happily call life.
I completely agree with that, and I think one can come out with very reasonable examples that are actually relevant to the origin of life. For instance, you may have noted that towards the end of the lecture I mentioned that we have no clear idea about the origin. We have a good idea about the origin of pores in membranes, about channels, but we have no idea how they actually originated at the beginning. But there are fascinating experiments done in the 1980s in which people took a very simple oligopeptide formed by serine, leucine. One of them was 27 amino acids long, the other was 14, and it was different numbers of serine and leucine all along the sequence. It's a normal peptide with no oligopeptide with no particular structure. If you put that peptide in contact with a bilayer, a lipidic bilayer, it immediately forms a proton channel-like structure that is telling us something that I think should be very relevant. All because of epistemological reasons, our understanding of the origins of life is based on a very Cartesian attitude in which we take the system, separate it in parts, and then try to synthesize the component. Nature doesn't work that way. So clearly, we need to go beyond that reductionist attitude, and that's one of the philosophical attractions that I have for complexity, but unfortunately, I have never seen it develop in full, but by others. So I think that is showing us that some of the traits that we are looking for with very complicated schemes may actually have a more simplistic answer if we then look at natural selection acting upon those kind of systems.
Okay, there was a question back there. Yeah.
Hi. I have two questions. The first one is, in this context of the RNA world, if we assume it's true, do we have any clue if all these catalytic reactions and the synthesis of biological components took place inside a membrane or just freely in the environment? And the second question is, if we have any clue if there could have arisen alternate systems of life, because you say that the ultimate common ancestor of life is posterior to the origin of life. So if there could be any other alternate systems that just because of natural selection got extinct.
There are fascinating experiments done. Well, the precursor of these experiments done between boundary defined system was Oparin himself. Remember that Oparin was trying to synthesize oligopeptides and polysaccharides inside quaterbates. Those became less and less fashionable, so the people have forgotten about those experiments. But on the one hand, Pierluigi Luisi and his group, now in Rome, he was at the ETH in Zurich, at the ETH in Zurich, and Dave Deamer in Davis, California, and Jack Shostak in Harvard, and most recently, Kepa Ruiz Mirazo from the Basque Country together with Robert Pascal in Montpellier, they have been demonstrated that some of these reactions are much more efficient inside the boundaries of a liposome than outside. So, and indeed you can actually promote oligomerization of a number of compounds like amino acids, ribonucleotides, and so on inside those compounds. And well, Robert Pascal told me a week ago, I was in his lab in Montpellier, he told me that they had very successfully demonstrated the charging of an amino acid to an RNA inside a liposome, and it was much more efficient than outside. The paper is now under review, so I'm sure I'm not doing anything wrong by quoting him. Now, the other question is alternative forms of life. Remember that when I described, I was very careful about that. When I described the nature of the last common ancestor very succinctly, very rapidly, I say it's based on life as we know it. All the life forms that we know on Earth share the same biochemistry. All the life forms on Earth share the same genetic code, basically. All the life forms on share the same basic molecular and metabolic traits in one way or the other. So I think that from an extraterrestrial viewpoint, if I may say so, life is clearly monophyletic. That doesn't mean that during the earliest stages of evolution, other life forms evolved that became extinct. We have ever discovered anything of the sort, and that doesn't mean that perhaps under different conditions you could have different forms of life. That is actually one of the driving forces between some of the excesses that my colleagues have are always making with astrobiology, and it's also led to a very, very embarrassing situation that I'm sure Rosie could be much more willing to discuss than me about those silliness of arsenic-bearing DNA and so on. But all the biochemistry we know is very, very, very common. So either nature is so deterministic that it's the only possible way, which I very much doubt, or we are stuck with one single form of life.
There was a question way at the back.
Thank you for your lecture. I have a question about the formation of amino acids. Did amino acids were formed here on Earth, or did they came from the meteorite crashes during the early formation of Earth?
I think both. I think that some of the original amino acids could have been formed on Earth without any problem. I think we have a variety of mechanisms. My favorite one is the Strecker synthesis, because we can actually quantify it very clearly. We can see the equilibrium with hydroxy acid. That leads us to a number of chemical assertions. They are very solid. But you can also have amino acids due to the hydrolysis of hydrogen cyanide. There is a wonderful experiment done in the 1967, 1968. It was published in the Journal of Molecular Biology by Jim Ferris and his group. They demonstrated that hydrogen cyanide polymerizes very, very easily. We know that quite well. And then when you hydrolyze the polymer, you can have adenine, guanine, you can have orotic acid, which is one of the precursors of the biosynthesis of pyrimidines. You can have that was later demonstrated by our friend, colleague here from the university, Alicia Negron Mendoza, that you could also have carboxylic acids. So the hydrogen cyanide polymer is a very, very efficient way of synthesizing other compounds. My impression is that most of the amino acids that we see in meteorites were formed via Strecker synthesis, which is a standard synthesis where formaldehyde, ammonia, you form amino nitriol, you hydrolyze amino nitriol, and that's it. You got it without any problem. I think most of them come from there, but given the abundance of polymers in a meteorite, I wouldn't be surprised that some of them were formed from the hydrolysis of hydrogen cyanide. But the basic point is that what you see on Earth was formed either on Earth and enriched by things coming from the outside. And let me say something that, if I may, that I think is very relevant. People are not aware, in general, of it, but one of the most fascinating results of a space exploration is a demonstration that an early intense collisional history of was typical of all the solar system. everywhere where you look in the solar system, there are evidences of collision. So it's not that the collisions were rare. It was just part of the process of condensation of the planets, moons, and so on. And that really, there was no place in the solar system that was spare of it. So the contribution of compounds from outside sources doesn't mean that we are re-advocating the panspermia hypothesis. We're simply saying that, yeah, it was part of the natural development of the Earth and other planets.
Thank you.
Sure.
Well, first of all, Toño, let me give you my warmest congratulations for the very nice talk you have given us to us today, as usual.
Thank you.
And I have 2 points or 2 comments or questions. The first one is, let's assume RNA form. So how the stability of RNA was achieved? Because RNA by itself today, it's very unstable in the absence of proteins. The ribosome is a very nice example. The ribosomal RNA is very stable because it interacts with proteins. That's the first comment, I would like to hear from you. And the second one has to do with what you said about the amino acids in the catalytic sites being, I don't know how to say it, a relic of very ancient, a very old situation. But to have these amino acids in the right position, the protein has to fall. And if the protein doesn't fall, the amino acids doesn't fit in the right place. So it couldn't be the other way around. First, in some way, proteins evolve, stabilize RNA, and then by a certain mechanism, which I do not have very clear, they have selected the amino acids in the right place in the active site.
Those are very, very good issues, Victor, as always. And this is not going to be an exchange of compliments, but the questions are very good. The stability of RNA. I have never worked with RNA myself directly, never, ever. But all my friends who do so tell me that indeed the half-life of the phosphodiester backbone is very small for a very simple reason. You have the OH group in the carbon, in the 2' carbon in ribose, and it attacks nucleophilically the phosphodiester skeleton, and then you break. I mean, that can be enhanced with the pH, temperature, whatever. It's a very, very common reaction. But people like Andy Ellington in Austin have been underlying the fact that you can, in some cases, have RNA going on for, depends on the amount of RNA you have, and you can have RNA going on for very long periods of time, and it can be stabilized even by folding. So that could be an answer, but I agree with you that the problem with the RNA world is that we are moving in an interface between what is purely abiotic and already biological, from my viewpoint. So I think that has to do with it. You're quite right that it can be stabilized by proteins, but remember that you can have equivalent, you can have proxies to proteins. For instance, you can stabilize RNA with clays. That has been shown very clearly. There is an old paper, it was published about 12 years ago by Enzo Gallori from Florence, showing that in fact you could enhance by orders of magnitude the stability of RNA and DNA just by binding it to clays. I don't think the clays that he was using were particularly relevant for the primitive Earth, but the demonstration is there and the principle is there. The other question may be related, and I think it's fascinating. I completely agree with you that in order to have enzyme-mediated biological catalysis, you need a very complicated three-dimensional structure, tertiary structure. However, one can have catalytic activity by very simple peptides, dipeptides, tripeptides. I have done those experiments myself. We published some of them on the polymerization of CAMP. I don't think it was very relevant prebiotically, it was abiotic. Just by enhancing the concentration of the peptides of histidine dipeptides. So you enhance the concentration and then you have a simple first order kind of reaction and the reaction can proceed. You could picture that kind of thing on the primitive Earth. But I find most fascinating what you say about the possibility of proteins starting not as catalysts, but starting as chaperones. Let me use the word right away. Let me take the word out of the closet and say, let's think of chaperones. And that's an issue that a number of people have proposed years ago independently. One of the groups was Anthony Poole, now back in New Zealand. The others were Luis Delaye and me, who actually suggested, we were scooped by a person that needs not to be named, but we suggested that perhaps the original role of proteins, of small peptides, was not catalytic, but they were stabilizing catalytic or functional conformations of RNA. And then Luis and I decided, we actually have to repeat that analysis. We decided that if that was the case, we could actually make catalogs of RNA binding domains, which can be seen which is something we can do quite easily. And just by looking at the traits of those domains to see which of them were older, under the hypothesis that the first peptides, biologically encoded peptides, oligopeptides, were actually acting like primitive chaperones. And then the addition of functional amino acids, catalytic amino acids like histidine, will actually lead to eventually to more complex catalysts themselves. For a long time, I was troubled with the fact that if we want to have RNA binding domains, peptides, you need to have lysine and arginine, and we don't see them in meteorites. And just until John Sutherland published his work a week ago, it was not clear we had a good synthesis. It's still not clear a good synthesis of arginine. But then Lauren Williams, who's a major expert on the structure of ribosome, gave us a wonderful solution. Again, going beyond this simple Cartesian model, he said, well, look at some of the proteins that bind to ribosome. Some of the proteins bind to ribosome using aspartic acid, but the aspartic residue is negatively charged. So in principle, you would say, no, it would not bind to RNA, but it could bind to a metallic ion, and the metallic ion, positively charged, could bind to RNA. So this means that indeed, you could have models, very reasonable models of early RNA binding domains that could have perhaps stabilized catalytic conformations of RNA. I agree with you. It's impossible to believe that all of a sudden you have a three-dimensional polypeptide that's catalytic and binds to different functional sites and so on. I completely agree with you.
We have time for two last questions, provided one of them is short. So I'd like to get one more question from a student in the audience. Hold on, with the gray sweater at the back.
Hello, I was thinking how important could be the daily or even seasonal variations in the environment on the primitive Earth, and how this could lead to promote or even prevent the formation of these primitive compounds?
It's a very good question. It tells you how relevant geologically our simulations have to be. Remember that the Miller experiment had a reducing atmosphere, an energy source, some water, and that was it. One of the possibilities that has been raised and done experimentally was, what about having wet and dry cycles in a small pond? We now refer to those Darwinian ponds as such in Origins of Life literature, in which you could have simply the accumulation, the wet and dry cycles that will promote formation of bonds between the precursors without any problem. And going to seasonal changes, there is a beautiful example that was proposed by Stanley Miller and Leslie Orgel, and I'm sure they had the tongue in the cheek when they proposed it. And this needs to be explained. Hydrogen cyanide dissolves very easily on water, and it's not simple how you can, it evaporates quite easily, so it's not simple how you could have a good concentration of hydrogen cyanide if you want to have adenine and so on. So they realized that the Vikings, the actual warriors, the actual civilization of the Vikings, used to enhance the alcoholic level of their beverages by using what is called eutectic freezing. In other words, you have a mixture, a solution of compounds that have different freezing points, and then the water freezes, and the Scandinavians are still doing that actually. The water freezes, and then you just collect very reasonable concentrations of alcohol that can be quite satisfying gastronomically, let us say. So they did, what about the same happening with hydrogen cyanide solutions in the primitive Earth during winter and summer cycles? And indeed, the experiment has been done very easily. You freeze the water, you concentrate the hydrogen cyanide, and then it spontaneously undergoes the polymerization to form adenine or the polymer. So indeed, the recognition of seasonal activities can have more than one significance.
Okay, you were going to be the last question, but
Toño, Victor has already stolen part of my question about chaperones. I was wondering whether RNA can be a chaperone too. Do you have evidence for that? As an activity, as being RNA as being a chaperone?
No, it had never occurred to me.
Maybe.
It never occurred to me.
But
no.
the other point that I would like to...
It could, I mean,
this is a question. It's a question for you. But the other thing I would like to address at this moment is the extant life, as we know it, has organizers, self-organizers. I'm talking in particular to cytoskeleton, for example. So obviously RNA and this equation RNA protein, as Kim originally asked you the first question, that there had to be something that selected for a particular ribose, for example, to become ribose and not be an open molecule. So there had to be, the extant life as we know it has networks. So these organizers, how can you visualize that life came to be without this? So for example, how tubulin or actin have these particular functions that organize the cell, or even in a bacteria, so that there are microdomains within this cell, and also most of even the ribosomes are pulled down or organized by these structures. So how can you, or what can you tell us about your vision on these particular molecules and how life could happen?
It's a very difficult question, and I have no answer for that. The only thing I can say about it, it's actually not my idea. It's an idea that has been, many people have toyed with it for several decades. If you look at a metabolic chart, at the chart of metabolism, you see that yes, everything is connected, but there are sort of compartments. And that, I don't mean compartments in the sense of having membranes. No, they are sort of a functional subunits, if you wish. One can look at at a particular functional subunit, the histidine biosynthesis, the one that I have studied with Renato Fani especially, and it's clear that one can picture a simpler version of histidine biosynthesis. His A and his F are duplications. You can have the formation of the imidazole group without his H gene, which is a glutamine transferase. So you can come up with less complex pictures of what may have been metabolic pathways once you already have enzymes, for sure. Now, the question is, when you start seeing the connection of one of those units with other, or those, units, let me call them, with others, in the case of histidine, it's very clear. It's connected to ICAR, the aminimidazole carboxyl nitrile ribotide to it purine biosynthesis. But they could exist in isolation as well if you have enough sources of ICAR. So it was just one single molecule that connected them. What may be a useful approach, and this is just, I'm thinking aloud, would be to look at the way parasites function. Because the parasites tend to lose a number of traits. And that's true for all kinds of parasites, either bacteria parasiting other bacteria or animals parasiting other animals. And perhaps if we look at the way in which units are lost, one could have some insight in what things are older than other and how they became connected. But it's a very, very open question that really has to do with the origin of metabolism and regulation. And people working with networks have achieved successful insights. The work of Lorenzo Segovia, for example, just to name a Mexican colleague that has done beautiful work in that regard, is an example. But those idea of subunits, it's the same that you find in polymerases, it's the same you find in aminotransferases. So I think nature works that way, apparently. But I'm afraid if I did something else, I will be quickly going into silliness. So that's about as much I can say now.
Well, I think you might be going into overtime.
Oh, okay.
I've been asked that we need to wind things up. I'd like to end with a round of applause for the audience for their excellent questions.